Hi everybody, thanks for your attention and thanks for inviting me. So I will present here ongoing research on population genomics of the Iberian lynx, a work that has been doing in collaboration with two other people, Begoña Martinez Cruz, a postdoc in my lab, and José Antonio Godoy, that is the coordinator of the Lynx Genomics project. So, first of all, I would like to describe the outline of my talk. I will basically divide the talk in four main blocks. The first one, I will introduce a little bit what's the problem of the species and which challenges is facing. Afterwards, I will briefly describe the approach that I follow to produce a genome-wide data set of SNPs. That was afterwards that I will use. I have used to perform several population genomic analysis, and finally, I will present the results of two different methods to reconstruct the demographic history of the species. One to reconstruct the ancient history between 10,000 and 1 million years ago, and a second one that will tell us a most recent history. As we've been listening all these days, during the last century, there has been a huge decline in species density throughout the world, and this is preceded by a reduction in population size in the population at the population level in different species. It's well known the fact that the main reason for this decline in species richness and population size in different species is due to human activities and is well correlated with human population growth. And for example, habitat fragmentation will reduce the population size of different of a species, and that will drive an increase in the inbreeding in these species, and the consequence will be a reduction in the potential to adapt to different changes in such populations. If the situation persists, the species will fall in this kind of vicious cycle known as extinction vortex. So, unfortunately, the Iberian Peninsula, we have several examples of mammals and birds that have compromised their probability to survive. And one of the most dramatic examples is the Iberian lynx. And in fact, the IUCN has classified the lynx as the most endangered species of felid in the world. It's known to be endemic from the Iberian Peninsula, and there are reasons to think that the differentiation occurred during the late Pleistocene, about 1.1 million years ago, from the Eurasian lynx. And the Iberian lynx found a refuge in the southwest of the peninsula, in the Iberian Peninsula, from the glacial peaks. So, during the last century, the distribution range of the species has been dramatically decreased, and Nowadays, there are only 2 remaining natural populations, one located in Doñana with about, Sierra Morena, with about 200 individuals, and the other one in Doñana with about 70 individuals. With such a reduction in population size, we will expect evidence for genetic erosion, and some other markers, like microsatellite markers, have shown evidence for high inbreeding and a strong differentiation among populations, and there's also a low nucleotide diversity present in mitochondrial DNA that is not completely neutral. So, in order to better understand what were the effects of the bottlenecks that has suffered this species, and to understand the population dynamics, genomics, and demographic history, the new NGS technologies have offered the opportunity to better study different aspects of the genetic aspects of the species. And it will provide with several tools for annotation and a better understanding of which variation is neutral or functional. So, the data we have used in this project is 11 deployed, 11 whole genome samples of lynx males with an average coverage of 26x. 4 of them are located in come from Doñana, and 7 from Sierra Morena. And these individuals were chosen in a way that they were the minimum related, with the minimum relatedness possible based on microsatellite data. And additionally, we also have the chance to analyze Eurasian lynx male data, and we have Illumina pair and reads with an average coverage of about 65x. We decided to use the de novo SNP calling approach called Cortex bar. That the main advantage that it has is that it doesn't need a reference genome to do the SNP and genotype calling, and also shows a low false discovery rate for SNPs and a high sensitivity for indels. So we can do, we could perform multi-sample SNP variant calling, and the inconvenience of this method is just that it has an 80% less sensitivity because it's a bit more conservative than mapping-based approaches. Okay. So in this way, we obtained 3 different data sets. One set of SNPs coming from an interspecific SNP calling that will include the Eurasian lynx and serve us to perform some cross-species comparisons. A second data set with use the 11 Iberian lynx samples that will allow us to, that was used for the population genomics analysis, and a third data set that will be SNPs segregating in Iberian lynx, where we can't really know what's the genotype in the Eurasian lynx. So that was used for recent demography. So in terms of population genomics, we found several evidences of genetic erosion in the genome. First, this PCA shows how Sierra Morena really differentiates quite well with, from Doñana using SNP frequencies. And also the fact that in Doñana there is low genetic diversity, because when we examine the first axis, we clearly separate both populations, but in the second axis, what we observe is intra-population variation. A second observation is the distribution of the Tajimas-D along the genome. In this case, we will, in a neutral case, we will expect that as most of the sites are neutral, Tajimas-D will present a value of D equal to 0. So we will expect that the average will be 0. But Tajimas-D compares two different estimates of heterozygosity and will indicate us if there is an excess of rare variants or common variants throughout the genome. What we observed, looking at the genome-wide distribution, is that the average is displaced towards a value of 1, and this is indicating us that there is excess of common variants in our dataset. And that's probably due to a population contraction. Another observation is the fact that for certain scaffolds, here's an example, we found extended, here I show the nucleotide diversity in the x-axis versus the distance, well, versus the position throughout a scaffold of 6 megabases. What we have observed also is regions of reduced nucleotide diversity, especially in Doñana, in red, compared to Sierra Morena, that could span about 4 megabases. To measure the intensity of, the amount of inbreeding at genome level, we use two different estimates. One is based in individual sites, and the second in segments. And the first one compares estimates the probability for each individual, the inbreeding coefficient as the probability of being homozygous by descent or homozygous by state. And another measure is looking at segments throughout the genome that are more than 1 megabase long and have contiguous sets of homozygous SNPs. Comparing both estimates, we have found a high correlation, as we would expect, and also a strong difference, a significant difference between both populations, with Doñana having the highest, an average inbreeding coefficient of 0.54, and also a higher number of autozygous regions throughout the genome. In terms of heterozygosity or diversity within this species compared to the Eurasian lynx, and also among both populations, we perform a calculation of the heterozygosity at genome-wide level. The way we perform this could be a little bit, it's a bit preliminary, and we counted the number of heterozygous sites divided by the total number of megabase pairs in the assembly. In this table, I show the different values obtained for the Iberian lynx and the Eurasian lynx, and we have, what is evident is that there is a 63% reduction or difference between the Iberian lynx respect to the Eurasian lynx. What I mean is that the heterozygosity of the Iberian lynx is 63% lower than the Eurasian lynx. And between both populations, we also observe that Doñana has 36% lower heterozygosity than Sierra Morena. But how how is this heterozygosity levels? How are these heterozygosity levels compared to other mammals? A recent study allows us to compare our estimates with different mammalian species, and what is surprising is the fact that the two species with lowest heterozygosity are two felid species: the snow leopard with 200 SNPs per megabase, and the domestic cats with about 100 SNPs per megabase. So, in conclusion, compared to the cat, we could say that the Eurasian lynx has 30% of the heterozygosity observed in cat, while the Iberian lynx has 10% of the heterozygosity observed in cat. That could suggest that it has been a loss of diversity compared with the common ancestor with cat. In terms of the demographic history, so we were also interested in examining the amount of linkage present in the genome. In this figure, I represent the correlation between two markers respect to the distance in kilobases. And to measure the extent of linkage disequilibrium, we estimated the point of 50% decay for the LD. That corresponds with the length, the distance between markers that match with the 50% of the maximum linkage disequilibrium volume observed for each of the populations. In red, I show the decay for Doñana, and in green, the decay for Sierra Morena. So in this manner, we could say that the linkage disequilibrium decay of Sierra Morena is about 270 kilobases, and for Doñana, it's about 10 times more. No, anyway, this could be an overestimation due to the fact that the sample size is lower for Doñana than for Sierra Morena. Compared to cat breeds, the lynx shows, at least for Sierra Morena, levels of linkage disequilibrium that are similar to the most inbred breeds. And for Doñana, we could say that the level of linkage disequilibrium, or the extent of linkage disequilibrium, will be between 300 and 2,300 kilobases. Well, but we would like also to know what were the reasons, or what happened in the past to observe the actual inbreeding. So we try to reconstruct the demographic history for the species using two different approaches. The first one allows to reconstruct for each individual genome, diploid genome, the demographic history, estimating the fluctuation in population, effective population size throughout the time, and is based in the in the level of homozygosity in different areas of the genome, assuming that recent divergence from the common ancestor will correspond with areas of increased heterozygosity where the genetic drift didn't fix the variants yet. In this figure, we represented the fluctuations of effective population size in the X versus the time in the y-axis. And what we can say from this figure is that there are two important, two trends. First thing I would like to highlight is the fact that all the Iberian lynx samples follow the same demographic history pattern, and there is a slight difference towards the end where the method lose resolution or power, independently of the population where they come from. And the Eurasian lynx shows a similar pattern, but always with a higher effective population size. Here we have observed that there is an ancient bottleneck about 800,000 years ago, and a second bottleneck about 200 years ago, and a third bottleneck pretty recently, and probably about 15,000 years ago. What is surprising is the parallelism between the demographic histories of both species. Probably because they were under the same selective pressure or very similar selective pressures due to the environmental changes, especially glaciations and reduction in the habitat range or prey decay. But what is reassuring is the fact that similar patterns have been found in other species, like the brown bear that occupies the same latitude as the lynx. So here we see that there are parallel histories also for two different clades of brown bear. Finally, I would like to highlight that the last bottleneck that we detect for the Iberian lynx at 15,000 years ago will correspond with a reduction in population size from about 3,700 to 2,500 individuals. In order to study, so we would like, as you see, the resolution for the PSMC approach reaches just a time of 10,000 years ago. So we would like to know what happened before these 10,000 years. And that's what Begoña has been doing. She used a different approach using a software called DARI that is a maximum likelihood method that allows to estimate the best model for first demographic model and reconstruct the history based on the allelic frequency spectrum. For that, we use a data set where we have the ancestral genotype for the Eurasian lynx. So, in this analysis, Begoña compared two different sets of models. One assumed that there was just one bottleneck, and also two alternative subsets of model, one with a drastic bottleneck, very sudden, and another one with a exponential decline in effective population size. The most likelihood model was a model with 2 instantaneous bottlenecks throughout the time, at different periods of time. First bottleneck dated 56,000 years ago, where the NE dropped to about 3,000 individuals, and a second bottleneck about 400 years ago, where the NE dropped drastically to 332 individuals. So, in conclusion, we have our analysis of the lynx genome shows a lower heterozygosity, 63% lower than the Eurasian lynx and 90% lower than the domestic cat, an extensive linkage disequilibrium decay, especially in Doñana, and a higher homozygosity in Doñana, supported by several observations. Long regions of reduced nucleotide diversity, extensive linkage disequilibrium, a 30% lower homozygosity than in Sierra Morena, and higher inbreeding coefficients for each individual in that population. And finally, our analysis of the demographic history allows us to understand 4 bottlenecks that occurred before the last century, and they happen at 4 different times, 800,000 years ago, probably due to the divergence between the 2 species of lynx and coincident with a long glacial period that happened afterwards, this long glacial period. A second one between 250,000 years ago. A third one between 15 and 56,000 years ago, where the population, effective population size diminished from 40,000 to about 3,000 individuals. And a last one, pretty severe, that was occurred 378,000 years ago. Nothing else. I would like to thank my collaborators and other members of the group at EBD CSIC, Estación Biológica de Doñana. The people at CSIC that has been sequencing the genome, they also assembled the genome. Some collaborators at NCBI that provided best blast hits to CAT. A researcher at CINIO that provided us with synteny analysis between lynx and cat to determine which chromosomes were autosomes and sexual chromosomes or mitochondrial DNA. The partners in the population genomics project and the funding agencies. And you for listening. Thank you very much.